So continuing with our lectures on synaptic transmission, we have zoomed in a little bit on the synapse and shown what an excitatory postsynaptic potential and an inhibitory postsynaptic potential look like because of the change in polarization at the membrane. What I want to do here is look at the difference between two different ways to sum up those excitatory or inhibitory postsynaptic potentials. And in this image, really we're looking at one of these types. So just really quickly as a brief review, I just want you to label this as an excitatory or inhibitory postsynaptic potential, right? It's always depolarizing, leading up to an action potential. Is that excitatory and inhibitory? It's one of your labelings to do. And then for the rest of this, I want you to look at the image and see how the difference is happening here versus what is happening here, and see if you can take the two of these and put them in the right spots just as a explanation of what is happening based on this image. And then if you can extrapolate from those definitions is which one of those is temporal summation and which one of those is spatial summation. So go ahead and pause the video and give that a try. So hopefully what you were able to see or at least think about is that when in this one, when you see the summation that's reaching up to the action potential, and really quickly, these are both EPSPs, so I'm just going to put them right in between there. There are no IPSPs, but just as a reminder that that depolarization is an excitatory postsynaptic potential, and these ones happen to be adding up to reach threshold. So you can see that in each one of these you have three action potentials that are releasing neurotransmitters onto this particular neuron. This one also has three action potentials releasing neurotransmitters, but in this one you have one neuron giving input to the cell. So this one is just adding up input from one neuron, but in quick succession, so that it depolarizes with the first action potential, depolarizes with the second action potential, and then further depolarizes with that third action potential, and by that time it reaches threshold. So this is the one that is summation from the same neuron over time. And then this one here is that you have input from three different neurons. So you get three action potentials from different neurons happening at the same time. So it is a faster depolarization that's going to reach threshold. So again, you have three action potentials, but they're coming from multiple different neurons. And so the way I think about this is that this one is you are adding up the action potentials over time from the same neuron. So this one has to happen first, then this one has to happen, and then this one has to happen. So this is over time, whereas this one, you are adding up the action potentials or the inputs over space. So the temporal summation, temporal just means time, so this is the addition of action potentials leading to EPSPs, wow, words, letters. So EPSPs are excitatory postsynaptic potentials over time from the same neuron. And then this one is you basically adding them up over space. So this one is spatial summation because you are adding them up across multiple different neurons. So now what I would like to do is I'd like to take all of these concepts that we have talked about across the last couple of chapters and put it into a larger context or a bigger picture so you can see how all of these things are interacting and affecting the signaling of neurons. So I have this image that is three different neurons. So this one cell body here with the dendrites, and it's receiving input from all of these cells. And then at number 2, something is happening. At number 3, something is happening. At number 4, something is happening onto this second cell. And then basically at number 5, we're starting back over at the beginning, but just within this cell. And then at number 8, we're ending it there, but then new things are starting within this cell. So just to recognize that there are multiple neurons within a chain for communication, and then helping you see how all of the things that we've already talked about tie in together to lead to neuronal signaling. So the first thing to do is to put basically 1 through 4 are all of these, and then just as a spoiler alert, they start over at 5 through 8. And then for each of those steps that is occurring there, I want you to think about what different type of channels are associated with those. So we haven't gotten quite yet to the G protein coupled receptors or the ligand gated receptors and the differences between those, but hopefully you'd be able to put them where they need to go because we have mentioned receptors in general. And then we've talked about these 3 different voltage gated ion channels and what role they are playing, but I want you to be able to see it with a big picture to put those details in where they go in the overall signaling. So pause the video and see if you can move these around to put them in the right spots for the particular aspects that are happening, and then also what different channels or receptors are involved at each process. All right, so we will begin at number 1. And so what's happening here, this is where your EPSPs and IPSPs, so those excitatory potentials and inhibitory potentials, those are being activated. So, you know, here is where you get the release of the neurotransmitter binding the receptors, but within this cell, the kind of first thing that's happening is the receptor activation is leading to inhibitory or excitatory postsynaptic potentials. And then at number 2, hopefully you were able to remember that this area right here, the transition between the cell body and the beginning of the axon, is called the axon hillock. And this is where all those IPSPs and EPSPs are adding up. And so that's the summation. And if it reaches threshold, then you're going to get an action potential. And the action potential is going to be conducted along the axon. So here's where your action potential is occurring. And then once the action potential gets down to the end of the axon, then that is going to trigger the release of the neurotransmitters into the synaptic cleft, and then those neurotransmitters are going to bind to the receptors on the postsynaptic membrane. Then at this point, you basically start over here, where those receptors being activated change the membrane potential and give you an inhibitory or excitatory postsynaptic potential. So you start all over again at number 5. Those will all add up. You may get an action potential if it reaches threshold, and then that will cause neurotransmitter release onto this last neuron here. And then in addition to all of those things, hopefully what you were able to see is that here is where we are talking about receptors. So that receptor activation is happening. That's what is causing those inhibitory and excitatory postsynaptic potentials. So technically, if you put your receptors here, you'll notice that this also ends in receptors. So you could put the receptors here as well. But I mean, basically that's the same, the neurotransmitter release binding the receptors and the receptors changing inside. That's basically all the same step, really. So you can either have it here or you can put it over here, but I like to put another one over there to save space. And those different receptors that can be activated are either ligand-gated receptors or G protein coupled receptors, and we'll talk about those in just a second. So once you get to the axon hillock, hopefully we're able to remember what type of channel is concentrated here in order to lead to the action potential, and that is the voltage-gated sodium channels. So though that concentration of those voltage-gated sodium channels is localized here and along the axon because this is where they are most important. So all of those postsynaptic potentials, they undergo this kind of passive spread until they get to the axon hillock, and if it's enough depolarization or excitatory postsynaptic potentials to reach threshold, then those voltage-gated sodium channels that are localized right here are going to open. That's going to depolarize the membrane, and then that is going to lead to the membrane depolarization. And remember, it's like that section of the membrane gets depolarized and has an action potential. That depolarization triggers the next portion of the membrane to have an action potential. That depolarizes the next. So it's this propagation of the action potential along the axon in little membrane segments. One thing that is not accurate is to say that a action potential propagates down the axon like this. That's sometimes what people think, but it's not. So not like that one, but it's just this little section of the membrane gets depolarized, then this one, and this, then this one, and then until you get down here. So that is, the depolarization is caused from the voltage-gated sodium channel. And then, if you remember, the other channels that, or the other voltage-gated channels that are responsible for the action potential, the depolarization is going more towards the sodium equilibrium potential, but then we have to have it come back down, otherwise we wouldn't be able to have another one, and bringing it down or going through that hyperpolarization is the voltage-gated potassium channel. So the action potential is really generated by opening and closing of the voltage-gated sodium and voltage-gated potassium channels. So that gets down to the end of the axon terminal, and the depolarization that happens down here triggers the voltage-gated calcium channels to open. And those voltage-gated calcium channels are what cause the neurotransmitter release. So big picture, including all of the different channels and receptors that are involved in neuronal signaling. So practice this. Make sure you understand all the little mini components that we have already talked about as well. But this is a good overall big picture view to help you see it in how it is affecting neuronal signaling. So at this point, I am going to zoom in on these ligand-gated receptors and G-protein coupled receptors so you can see the difference between those. We will get into more specifics on them when we talk about the specific neurotransmitters and their specific receptors, but I want to focus on the overall general differences between the two. So when we are talking about ligand-gated ion channels, we are talking about channels that are opened and closed based on the binding of receptors. When we talked about the voltage-gated channels, then we were talking about channels that were opening and closing based on hyperpolarization or depolarization of the membrane. So a charge-based opening and closing versus a neurotransmitter or ligand-based opening and closing. So in this, the terminology, the ligand here, ligand is just something that binds and activates a receptor. So the ligand here for us, and when we're talking about the synapse, is a neurotransmitter. So you can also consider these neurotransmitter-gated receptors, but the generic term is ligand. So when the neurotransmitter binds to the receptor, you can see that it opens a channel, and then in this, it's associated with an ion channel. So ions are going to move across the membrane depending on where the concentration difference is. So sodium and calcium and chloride are all going to move from outside the cell to inside the cell. If it is a neurotransmitter channel that is permeable to potassium, then the potassium is going to move out of the cell. But there are many different ones, and we'll get into the specifics of different types. But the main point is that in this one, the neurotransmitter binds a receptor, opens an ion channel, ions move through. When the neurotransmitter leaves the binding site, then the ion channel closes. End of story, nothing more. The G-protein coupled receptor is a little more complicated, and I've drawn this circle around here because in this image, this is the same receptor, it's just closed or open. In this one, this receptor is different than this particular channel. This is just something, the G-protein coupled receptor is affecting this channel. So it can affect ion channels, but for the G-protein coupled receptor, it is not directly associated with with an ion channel. So when the neurotransmitter binds to a G-protein coupled receptor, what happens is, again, the neurotransmitter is able to cause a change in shape, but since this receptor has no ion channel, something else has to happen. And what happens is the activation of a G protein. So the neurotransmitter binding changes the shape of the receptor, which then changes the interaction with the G protein, which then changes the shape of the G protein. And the G protein is able to go and then activate usually another enzyme. And then there are a couple different things that can happen here. It can either then go and affect an ion channel, or it can go and affect other intracellular signaling cascades that will activate one thing, which will activate another thing, which will activate another thing, which will then go do some kind of signal, like intracellular signal change. And one of those changes can actually be DNA transcription. So you can change gene expression based on G protein coupled receptors. So a longer chain of events that happens, and then it can change things that have a longer change within the cell. So what I want you to think about is, what are the differences between these, just based on that quick introduction? What do you notice based on the picture, and what do you notice based on the explanation that might tell you something about, this one's a little bit easy, like what's it associated with? But it's important to recognize how the name relates to part of its function. And then one that's a little bit more difficult is which one has multiple subunits and which one has one subunit. Maybe you can tell from the picture. And then something just in terms of how fast the signal happens or how slow it happens. And then whether the effects are long-term or short-term. So go ahead and pause the video and see if you can come up with those just based on that explanation. So I'll start with the easy one. The one that is associated with an ion channel is the ligand-gated ion channels. And the one that is associated with the G protein is the G protein coupled receptors. So hopefully that's a little bit obvious. And then the ones that are a fast signal speed, if you happen to remember, I said that when the neurotransmitter binds an ion, the ligand-gated ion channel, it opens, and then you get a change in membrane potential. That's a pretty quick signal. So that has the ligand-gated ion channel is a fast signal speed because it opens, and that's the response. The G protein coupled receptor, in contrast, the neurotransmitter binds to the receptor activates the G protein goes to activate an enzyme, and then it can either then open the ion channel, but it takes longer, and then it can also, sometimes the effects are more downstream, which can take even longer. So it often has a slow signal speed. But along with that slow signal speed is that once this gets activated, and then it activates this, and then it might activate something else, which might activate something else, which can then even go and act and change gene expression, right? That can be a much longer effect. So even after this neurotransmitter has left the receptor, the effects within the cell are still ongoing. So it has longer term effects. So it has a slow signal speed, but the effects can last longer, and sometimes, depending if it actually goes to affect gene expression, the effect can last days or even weeks. And then, in contrast to that, the ligand-gated ion channel, it opens quickly, right? But then once the neurotransmitter leaves, it closes just as quickly. So the effects are very short term, because once the ions move inside or outside, right, then some kind of pump is going to reestablish the membrane equilibrium relatively quickly. So the overall timing of the effects for the ligand-gated ion channels is very short. Now the one that's a little bit more difficult to get from this, if you haven't had like a lot of bio previously, is the multiple subunits versus one subunit. You might have been able to tell from the picture that this is one chunky subunit. It's one piece. The ligand-gated ion channels are often multiple pieces, so you can kind of see that there are multiple segments that make up the channel here. So a ligand-gated ion channel is multiple subunits, and a G protein coupled receptor is one subunit. And then I just drew this down here to kind of show you why that is, because here you can see, if you're looking from the top, right, you can see that there's this subunit, and then this one, and then this one, and then this one. So this one has 5 different subunits. If you look at this from the top, what you would see was this. If you had a subunit here, a subunit here, a subunit here, and then what those subunits coming together are creating are a pore in the middle of that so that the ions can move through. So this is why these are ligand-gated ion channels, because there are multiple around there, and those multiple coming together create a pore in the middle. Whereas the G protein coupled receptors, it's just one subunit, and so it's not bulky enough to really create a channel or a pore for ions to move through. So the G protein coupled receptors are only one subunit, whereas the ligand-gated ion channels are often multiple subunits, generally like 4 to 6 subunits. And that completes our discussion on synaptic transmission. Our test is coming up. Go back and check out the objectives review. And if you have questions, again, submit those for Tuesday's class. And then if you need more help, then see me outside during office hours. I had sent an announcement, so check out that announcement if you need details on my office hours. But I do recommend, if you have questions, or if you're specifically an auditory learner, sometimes it helps to talk things out. So contact me if you need any help getting ready for the exam.